All right, good morning, everybody. Happy Monday, as they say. Let's get going. All right, so we were basically finished up about here on Friday. So let's put the chat over here. All right, we ended up talking about replication fidelity, right? How accurate the process of DNA replication is and what leads to that accuracy, right? So there's a few different processes and characteristics that give us our accuracy, which are summarized here. So overall, this is like the summary. So essentially what we have left to do is we need to talk about DNA replication in eukaryotes, essentially the differences between prokaryotes and eukaryotes. We need to talk about the end replication problem, which is pretty interesting. And also we need to talk about homologous recombination. Now, I don't think we're gonna get all of that done today in the next 50 minutes. So we'll probably finish that up on Wednesday. And hopefully we'll have time for, a little bit of question and answers, clarifications, maybe even practice some 3-point crosses, stuff like that, right? Because I know it's not 100% for everybody, 3-point crosses. Oh, I'm sorry. I got woken up by the cats doing something stupid in the middle of the night, which is pretty common. Okay, so essentially, we have a whole bunch of stuff, right? And so this is where the challenge in kind of this part of genetics moving forward comes in. Is it's just, oh, the dog coming down. It's just, well, it's not just, that's not really doing it credit, but it is a bunch of stuff to learn, right? Because all of these processes are complex, right? Even in the form that we give you as a somewhat of a simplification of all of this, right? But there's a lot of stuff going on, right? And so some people kind of learn this best just by brute force, right? Kind of like pounding it into your head, things like that. That's fine. Personally, I tend to learn best by understanding why stuff does what it does, right? The actual kind of meaning behind it, I guess, right? And so that's what I would suggest to you. Obviously, you use whatever study method works best for you. But understanding what all these different things do makes it a lot easier to put them all together into a bigger picture, right? And as always, right, I'm going to ask you questions in the exam, which will test your understanding of this from multiple different perspectives, right? I'm not just going to get you to regurgitate what I've told you in lecture, right? Because that you wouldn't necessarily understand it if you were able to do that. Okay, so essentially we've got our topoisomerase, relieves supercoiling, single-stranded binding proteins, and helicase are involved in opening up and keeping open the replication bubble. We have 2 DNA polymerase complexes which are actually linked together, right? They are not kind of like floating around in the breeze, right? And that means that DNA is synthesized at the same rate for both lagging and leading strands. We've got our RNA primase here. Again, this is in prokaryotes, right? In eukaryotes, actually one of the DNA polymerases is also a primase, which is pretty funky. We have other DNA polymerases, again, in prokaryotes or DNA polymerase, that removes the primer, and then we have DNA ligase. It's just my little dog goes down the stairs by like, which is always makes me laugh. And we have DNA polymerase, which seals the backbone mostly in the lagging strand, but also in the leading strand too, right? Because even if you have unidirectional replication in bacteria, you know, you're still going to have one long linear piece of leading strand, which needs to get stuck back together. Right? It just happens much, much less frequently, even in eukaryotes, than in the lagging strand. Okay, so moving on. Actually, before we move on, anybody got any questions? There's one of my other cats. Let's see if you can see her. She's like a snow leopard. Never ever get to see her. Where is she? There's the stairs. I don't know if you can see her next to, behind that column. There you go. Jasmine's coming down, love. Yeah, my wife said, wow. Okay, anybody got any questions? Stuff that needs clarification or repetition? No? Okay, so moving on to eukaryotes. Right, so by and large, the principles are the same. Differences come a little bit in some of the processes and in what does those processes, right? So kind of one of the big points is that eukaryote DNA is linear, right? Obviously, apart from mitochondrial DNA, which is kind of not eukaryotic, I guess. And as a consequence, and it's also really, really large. So as a consequence, you also have multiple origins of replication too. So that's kind of one big difference. The other big difference is that eukaryote DNA is always complexed with proteins. And so eukaryotic DNA exists as chromatin. And chromatin is DNA plus histones. And these histones are used to package DNA to allow it to fit in the nucleus and also control the access to that DNA. So it's actually a really big part of control of gene expression, which we'll talk about later on in the course. Now, we're not going to get into that. We're just really going to introduce the kind of idea of histones, which I'm pretty sure should have been covered in GenBio 1. But if they weren't, we'll cover them pretty thoroughly here. And there's also, you know, differences, as I said, in like what does what and how it does it too, which we'll get into. Before we get into that, though, we need to talk about chromatin, right? So I don't think I've introduced this before. Essentially, DNA exists in eukaryotes wrapped around little balls of protein complexes, right? And those balls consist of 8 histone proteins or 4 pairs of histones. So you can see the different numbers here. Don't worry so much about the numbers. These numbers are actually really useful in terms of the histone code, right? And so different histones get modified in different ways in different places. And that generates, you know, different behavior essentially in terms of the protein. And these balls are called nucleosome core particles, right? And so they look like beads on a string, basically. And that's where that term comes from. And they're held together and spaced apart by another protein called histone H1, right? So about 1 nucleosome core particle and the DNA between them is usually about 200 base pairs, give or take. I'll just write that down for you. So that's the nucleosome core particle is 8 histone proteins plus histone H1. Oh, actually, no, let's put it a different way. Plus around 147 base pairs of DNA. plus linker DNA, which is the bit in between, which allows these things to kind of move around and wrap around each other, plus histone H1 equals a nucleosome. And that is around 200 base pairs of DNA, give or take. Right, so I always have to try and be really careful with language here because it can be confusing. So the core particle is just the bit of DNA wrapped around those proteins. But there's also a little bit of DNA in between each core particle to allow basically to allow them to wrap around each other. Right, and also to kind of shift around too. There's a lot of movement and so on and so forth. And so from the beginning of one nucleosome core particle to the beginning of the other, that's about 200 base pairs. That's often referred to as a nucleosome. So there you go, that's a good definition. And eukaryotic DNA typically exists in the form you see on the right, right? This is a kind of standard chromatin view, right? It can get more compacted than this. So this is what we would call euchromatin. Ooh. Jasmine is chatting. All right. So that's chromatin, right? Oh, and so one of the challenges, obviously, is in eukaryotic DNA replication, you have to kick these off, right? So because otherwise they'll get in the way, right? So you have to kind of like shuck them off the DNA as you go along, and also you have to replace them, right, afterwards. So once all of DNA replication and error correction, like mismatch repair, stuff like that, methylation has occurred, you need to wrap that DNA back around histone proteins. And so because you have twice as much DNA, you also need twice as much histones, right? So there's an awful lot of energy spent just making the histones to wrap that new DNA around. Also, it's kind of like, on top of that, these histones have markers or marks, right? It's called the histone code, right? Which alters their properties and their behavior. And so when you replicate your DNA, you're going to have roughly 50% old and 50% new histones, right? So if that DNA replication is leading to meiosis or as part of meiosis, right? Essentially, the offspring are going to have half of the marks of one parent, which is kind of cool. Yeah, so it's actually, that's right, Caitlin. So it's kind of a complex concept to give. It's basically, chromatin is in a constant dynamic state, right, between being open and being closed. And what I mean by open and closed is basically switching between an accessible form of chromatin, which is called euchromatin. So, oops, if I can spell it right, equals open chromatin that's accessible, oops, if I can spell that right, for transcription. Yeah, it is spelled that right. Whereas heterochromatin is closed or shut down, equals no gene expression. I don't know what my dog's doing upstairs. So some parts of the chromosome are always shut down, right? They're never transcribed. There are very few genes there, if any at all, like the centromere, for example. But the rest of it exists in this dynamic state between open and closed. And the reason for that is you don't have enough space in the nucleus to have all of your DNA in the open form, right? So you have to It's like those, I don't know if you've ever been down into the basement of a large library, like a large university, or I think even Central Library in San Antonio has this. But they have those roller shelves, right, which you can kind of roll up, get the thing out that you want, and then hope that you get out before someone else needs something somewhere and squishes you. They're really, really cool. I don't know why. I always love them. It's kind of like that, right? So you only have a certain amount of space, and cool. And you can only access part of it at any one time. So because needs change over time, like daily or developmentally, you basically open and shut different areas of the genome as needed, right? So that's a very big part of controlling gene expression. Hello, doggo. Luna dog has decided to get out of bed. This is our teenage dog. So yeah, it's kind of how it's stored, but that doesn't really capture the full impact of what chromatin is about, right? It's how it's accessed. It's basically how you solve the conundrum of fitting a lot of DNA into a very small space, but being able to access it. There's also sorry. Oh, it's so funny teaching from home. Where was I at? Concentrate. Yeah, it's also a way of controlling gene expression on a very broad scale, right? So there are things called developmental programs, like, if you want to develop an arm, well, actually, you typically want to develop two, right? There's a whole bunch of different proteins or genes which are involved in that. And then once you've got arms, you've absolutely no need for those proteins anymore, right? So you can actually shut down like a whole swathe of a chromosome, which is involved in a particular process, which is kind of done with, essentially. So anyway, that's kind of, it's a very, very, oh man, there's so many details with chromatin. Like the histone code is like another language, one I don't kind of sort of understand, but not super well. It's not really my strength. But yeah, it's actually a very, very interesting topic. It is. It's on the techniques that people have developed to visualize that. Like you can actually look at changes in chromatin state live in live cells using fluorescence microscopy. So you can actually look at the nucleus of a cell and you can see kind of like different areas of the nucleus flash as it kind of gets turned on in terms of gene expression, and then it kind of goes off as it gets silenced again. It's really pretty cool. Okay, so moving on from that, in where are we at? Let's go back up here. So this is what DNA polymerases look like in E. coli. And sorry, got to focus. We have one DNA polymerase which does it's like the main workhorse. That's DNA polymerase 3. We have one which does just primer removal and gap filling, and then we have a few others that do DNA repair. So in eukaryotes, this is typically the case, right? They have lots and lots of DNA polymerases. And the key difference here is that we have a starter DNA polymerase and a different DNA polymerase for the leading and the lagging strand. So the starter DNA pol is alpha. And that basically takes the role of primase. So I'm guessing, I actually don't know, but I would guess that there was some kind of gene fusion, right, in the past between a DNA polymerase and an RNA polymerase or primase. So you ended up with one protein which has two different kind of domains and functions, right? So it's both primase and synthesizes the first, I think it's about 30 base pairs, give or take, of both. Oh, I'm very sorry to hear about that. Let me know if you need any help. Sorry, someone just has to leave. So yeah, my condolences. Okay. Yeah, as I said, all this will go up. If actually send me an email later and we'll talk about like the exam and stuff. Okay. So don't worry about any of that right now. Go and do whatever you need to do. And I'll talk with you later. Okay, so yeah, it does both leading and lagging strands. But once it's done a little bit, it falls off, right? And it's replaced by a strand-specific DNA pol, right? And so in, oh shoot, what is that letter? I hate bloody Greek letters sometimes. I'll look this up. Hello, Troubles. We have more. Pepper. Oh, you can see my feet. I hope none of you have a problem looking at feet. And there's the other one. She's a real pain in the ass sometimes, that dog is, I have to say. Oh, I'm getting warnings from my computer. Let's close down this. Probably because of Gazonga's amounts of tabs I've got open. All right, so I need lowercase. There we go. Yeah, they're very funny. Oh, delta is the funky one, and that's epsilon. There you go. I'm going to type that into the notes because I'm guaranteed I'll forget. And epsilon, which is like the Greek E. All right, anyway, so delta, pardon me, does the remaining part of each Okazaki fragment, which isn't actually all that much if you think about it. They're only 200 to 300 base pairs long in eukaryotes. So once DNA pol alpha has done its job, there's like, you know, only a couple of hundred bases for DNA pol delta to do. Whereas DNA pol epsilon does the whole of the leading strand, right? So that's a big difference, right, between prokaryotes and eukaryotes, right? The DNA polymerases are much more specialized. And also, the process of removing primers is quite different, right? So if you remember at where we're talking about prokaryotes, it's all done by DNA polymerase 1. Right, so DNA polymerase 1 both removes the RNA primer and adds in the nucleotides to fill that gap that removing that primer creates. So in eukaryotes, it's quite different. So here what happens is essentially DNA pol delta, right, which is doing the lagging strand. Essentially, it kind of keeps going until it butts up against the beginning of the RNA primer for the previous Okazaki fragment that was made. And so it doesn't have any nuclease activity of its own in terms of removing the primer. What it relies on is another protein, this replication protein A, to flip out the RNA primer. I don't actually know how it does that, kind of gets in there and who's out? Oh, you're back again, are you? Okay, behave yourself. As long as they're not going gonzo with each other, we should be good. Oh, you need to come up a little bit closer. Oh, this is the best. Isn't she adorable? We call her the Franken puppy because she's had so many surgeries and she has these scars all over her chest. So we're thinking about dressing her up as Frankenstein's puppy for Halloween. Not entirely sure how we do that, but yeah, she's very cute. Super loving Gonzo. They're probably just hanging around wondering why I'm not taking them for their morning walk, most likely. Anyway, so the RNA primers flipped out, and then replication protein A brings in an endonuclease. So remember, we talked about endo versus exonucleases. So endonucleases cut within DNA or RNA actually. And exo, come on dog, get comfortable. Exonucleases cut the ends of DNA, RNA. And it's a really important distinction between the two because, well, it depends on what cuts what and where, essentially. So anyway, the replication protein A recruits endonucleases, and what they do is they cut off that flipped out piece. And so now you have essentially kind of an open section of track ahead of DNA polymerase delta, right? So you removed the obstacle that was in its way, and it then just continues until it hits the beginning of the next piece of Okazaki fragment, but the DNA of it, right? So now all that's left is a gap in the backbone between the sugar of the last nucleotide added and the phosphate of the next nucleotide. And that obviously gets sealed by DNA ligase. The bit that was broken out is just digested by exonucleases. Typically, whenever you see bare DNA, like single-stranded DNA or RNA floating around in the cell without any protection, right, on the ends, which we'll get to when we talk about transcription, it gets cut up. And part of the reason why it gets cut up is because you want to recycle all the goodies, right, that it represents. But also, it's part of the cellular defense mechanism against viruses. And so if you find single-stranded DNA or just, unprotected RNA flown around, there's a good chance it's not yours, right? So you want to get rid of it. Okay, so one of the, in my mind, kind of coolest things about DNA replication in eukaryotes is what we call the end problem, the end replication problem. And so what I'm going to do is, if Doc will let me, I'll actually draw this out because it's really quite kind of crazy. And this is actually one reason why you get old. Obviously, you get old because you live long enough to be older, right? Kind of self-evident. But it's all right. Nothing's happening to you. All right, we're going to have limited amount of board space due to dog draped over my leg here. Ain't she? Sniff, sniff. All right, here we are. I remember giving one of these to her as well. She actually got it as a puppy. Chewed the end of my marker off. Didn't you, doggo? Black, just like you. Anyway, Okay, I don't think she likes the smell of those. It's all right. It's not going to do any harm to you. Use it to color in some of her white patches, so she's not so spotty. Okay, so the issue is the ends and the primer. Right, and this also comes back to the whole point of needing a 3' OH to synthesize DNA, right? So all of those things kind of come together to create this problem. And so, what are you getting excited about? If you funny dog. Right, so this is the template DNA. Oh, I'm gonna need, and let's just make it simple, right? So this is 5'. So you're going to need a primer here. And you're going to need one here. Right. We're going to ignore the fact that there's going to be multiple points of multiple replication bubbles for now, just to keep things simple. And so DNA pol comes in and so funny, synthesizes the DNA to match, right? Let's just do that for, let's say, the bottom strand, right? We'll repeat the synthesis of this. So now we're going to get rid of the RNA primer, right? Because RNA primers, well, RNA is not part of our genome, right? And it shouldn't be there. It causes all kinds of issues. So when we replicate that, essentially we're going to have, oh, actually, let's do both of them. Let's see if I can avoid this from being too confusing. You'll have this, oops, I don't get the couch dirty. This situation, right? Where each of these, so red is basically the new DNA, so that's why it's all blue here. And you're going to repeat the whole process again. So you're going to have another RNA primer, but now you have an RNA primer here again. But now because your RNA primer was removed and it can't be replaced because there is no 3' OH here. Does that make sense? I think I skipped over a little point. So basically, there's no way DNA polymerase can fill in this gap. Any that are in here are going to get filled in by DNA pol delta, for example, because there's a 3' OH at the end of this Okazaki fragment to use to synthesize the strand to replace that RNA primer with. But at the ends, there is no bit of DNA because it's at the end of the chromosome, right? So what that means is that every time you replicate your DNA, right, we have the same going on here, right? So imagine this is, you know, cells that are going through repeated rounds of replication. your DNA gets shorter. And it gets shorter, or each chromosome gets shorter at each end by the length of one primer. So you can see this copy, chromatid, I guess, future chromosome, is a little bit shorter. This one is going to be even shorter than the original, and so on and so on and so forth, right? This is called the end replication problem, right? And so it's dealt with in a couple of different ways. One is we have buffers, right? So essentially what you don't want is for these gaps that are created, these shortened bits, to start eating into coding regions. So if you have a, as a result of the end replication problem, you remove part of a promoter or part of a coding sequence, then you lose the function of that gene, essentially, or it may be expressed inappropriately and cause other problems. So we have basically buffers. Sorry, we've got a bit of a wobbly little table that I've got my laptop on. Anybody know what those are called? What are the ends of our chromosomes called? It begins with a T. We have centromeres at the middle, or a centromere. What's it called as stuff at the end, that feature or area or region? It begins with a T. Yeah, awesome. Cool, thank you everybody. You've saved me from what would be a rather torturous analogy involving telephones and what's in your pocket and stuff. Exactly. So telomeres, or telomeres, however you pronounce it, basically what they are stretches of repetitive nonsense DNA, basically. And this is a fun fact. My chromosomes are shorter than my daughter's. And my dad's chromosomes are shorter than mine, right? And so your chromosomes shorten as you get older, right? As that buffer is used up. So that buffer gets shorter every time your cells divide. So what do you think happens when you run out of buffer? What do you think the consequence is? Does it just keep on going and then you just take the risk? Or what else might occur? Remember, this is happening at cell by cell level too. Well, I'll tell you. That cell stops dividing. So you have about 60 cell divisions worth of buffer. This is called the Hayflick limit. And basically, once that limit gets hit, or really the buffer gets used up, not bugger, buffer. Cell stops dividing. And that is a process called senescence. Senescence, AKA old age. So basically your cells from birth have, or most of them, right, have a built-in cell division limit, right? after which they will stop. And actually, typically those cells then become kind of crappy and useless, right? And they get old and they stop functioning properly, and some of them end up being killed. Right? Others just hang around being like cantankerous pain in the asses and cause all kinds of other physiological problems. And that's a very big part of why you get old, right? And so essentially, the older you get, the more of your cells hit that senescence, that Hayflick limit, and go into senescence and stop functioning as well as they should do. So your overall physiology kind of gets worse. A little bit depressing to talk about, to be honest. Hey, cool. I'm glad about that, Melody. So chromosomes, or all eukaryotic DNA, exists in the form of chromatin. And chromatin is basically DNA wrapped around histone proteins, Caitlin. And so that DNA is both active and silent. And so it's both being used and being stored often on the same chromosome, right? It's not by a chromosome basis, it's by a region of DNA basis, right? Even down to, 100,000 base pairs or so. Yeah. And so that is a kind of dynamic state between open and expressed and closed and silent within the same nucleus, within the same chromosome, right? So constant dynamic switch. And so really the only time it all gets shut down is during M phase, right? That's what prophase, or part of prophase, is all about, is turning all of your DNA into heterochromatin. So it all just goes like that. And it's totally shut down and inactive, right? So that's why you get all of your kind of work done as a cell in interphase, because as soon as you enter prophase, you can't make any proteins. You have to have them already, essentially. So getting back to the Hayflick limit and telomeres. Can anybody think of cells in your body which do not obey the Hayflick limit? Good morning, love. What cells have to avoid the Hayflick limit for you to keep functioning as an organism, right? Because 60 rounds of cell division is not very much. Brain cells, actually, that's actually a good point, Isaiah. Brain cells are the ones that never divide again. Neurons are what they call post-mitotic, right? Once they've become neurons, they never divide again. Think about other parts of your body which needs to be refreshed, I guess. If I were to kind of go like this, clean off my skin cells, right? Because your skin cells are constantly dying and being replaced by, skin cells underneath them. That's why you get dandruff. What else? What other kinds of cells? Just think about what you're constantly replacing. Saliva? Yeah, saliva is more of a extracellular secretion, to be honest, kind of mucus and stuff. What about hair cells? Right, blood cells, exactly, white and red, although red are a little bit weird because they're kind of like a one-shot deal, basically. They don't divide again. Gut cells, right? So your intestine is constantly being rejuvenated, and a few other bits and pieces, right? Liver cells as well, if you've got an alcohol problem, stuff like that. So they have a mechanism, basically stem cells and germ cells, right? They escape the Hayflick limit by the use of an enzyme called, unsurprisingly, telomerase. And what telomerase does, and it's actually kind of funky, I only partly understand it. It uses RNA and other stuff. Essentially what it does, though, is it rebuilds the ends of the telomeres. And so germ cells like to make gametes, and stem cells like the ones that are used to kind of renew your skin or maintain your gut functional and stuff like that. Cool. Well, I'll take them out for a walk when you get back as well then. Yeah. Dogs are going to get a walk. See. Very alert and excited. Basically, those escape that limit, right? So they can essentially divide for the life of the organism that has them. And that's also one reason why if you get a high dose of radiation, where you tend to see the problems first are in the skin and the guts, right? Because those are regions of your body that have fairly active cell division. Okay, so just to end up, and this is just to kind of, you know, put another kind of worm in your head in a sense. What other types of cells, right, which you necessarily don't want, in fact, you really don't want them, are essentially immortal and can divide over and over and over and over and over and over again? Anybody think what kind of cells fit that criteria? Actually, the main cell line that most biomedical research has been built on, the HeLa cells, are from a lady called Henrietta Lacks. Does anybody know what kind of disease Henrietta Lacks had? Before, that's right, she had cancer. I think it was cervical cancer or uterine cancer, something like that. And before Henrietta Lacks came along, and there's a lot of kind of ethical controversy about this because she didn't truly have informed consent about what was going to happen with her cells. But before HeLa cells came along, you could only work with cells in cell culture for a limited amount of time before they died, right? Because of this Hayflick limit problem. And so her cells, which are still in use and are the foundation of most of the drugs that you take, for example, were immortal. They never died. And they never died because they had activated telomerase, so they escaped the Hayflick limit. So tumor cells basically find a way around this issue. Because if you think about it, if the Hayflick limit stayed in place for tumors would never grow beyond a certain size, right? Because each and every one of the cells in them would have a finite number of cell divisions before they senesce. And yet tumors can grow as big as the people that host them. And the reason for that is that telomerase is activated inappropriately in those cells, which is kind of cool. And so it also kind of gets back to this whole, give me a second, Isaiah, desire for longer and longer lifespans, right? That drives a lot of biomedical research, at least like the fancy stuff, right? And so you have to think, well, yeah, I mean, we can make ourselves live longer, but how do we do that without like unintended consequences such as cancer? So Isaiah, a question about hang on a second. Chapters in the oh, you mean PowerPoints? That would be a definite yes. You don't want to see my taxes. That is absolutely no use whatsoever. I just got to, actually, you know what? This is a better place to look from. It's Monday, Wednesday, Friday. And yeah, so the exam will be over everything from non-Mendelian patterns of inheritance, which is PowerPoint 7, right? So sex determination, sex linkage, then other forms of non-Mendelian inheritance, linkage, DNA structure, and DNA replication. That is what the exam will be over. And we will also finish up homologous replication on Wednesday. But I find this whole stuff about telomerase and cellular senescence and stuff so fascinating that I've just spent a little bit of extra time on it today with you. There will be no lecture on Friday, but I will be in the Zoom. So I'll be online in this Zoom session. If you have any questions or problems or last minute, how do I do this kind of stuff, I'll be there for probably most of the 50 minutes, to be honest. And I'll be available for the rest of the day as well. But there will not be any formal lecture on Friday. It will be the exam. Cool. Well, thanks very much, everybody. I shall see you all either tomorrow or Wednesday. Thanks. Have a good day too.